So, Dr. Mahad, I just wanted to initially really ask you if you can provide some of the background on how you and Dr. Nancy became involved in this particular aspect of progressive MS research and study.
Yeah, sure. So we've been working on progressive MS for the last 10 years. We started, so my background is I'm a clinician scientist. I trained at the Mellon Center doing a National Multiple Sclerosis Society sponsored physician clinical care fellowship. And then I got my a senior fellowship, and now I work in Edinburgh in Scotland. So we've been kind of both from a clinical perspective and a basic science perspective, been very interested in progressive MS for the last 10 years. Our approach was really to look at autopsy tissue from cases with progressive MS and to see how the energy producing factories, if you like, called mitochondria, the powerhouses, are really affected in progressive MS. And to our surprise, actually, we found quite a few changes, and most of these changes are in neurons. This kind of the cellular compartment, the neuronal compartment, is something that's actually received very relatively little attention in MS. One thinks of MS as a demyelinating inflammatory disorder, so you think about the myelin-forming cells, the inflammatory cells, and the neuron is very much regarded as the target of all these processes that damages. Now what we realized, the work done by us and many other groups now, is actually the contents within the neuron are changed quite dramatically, and one of these aspects is damage to mitochondria. And there's two essential observations I think we've made. One is when you look at the neuron, the cell body of the neuron, and look at the mitochondria there, they've got lots of abnormalities where the ability to produce energy is compromised. Okay, and then if you then look at the nerve fiber, the axon, which is kind of demyelinated in MS, you have an increase in energy demand. So when you lose the protective sheath, there's some changes that goes on with the ion channels, and you need more energy to just maintain the structure and function. So when you combine the two things, it becomes quite a unique situation in progressive MS, where the ability of the nerve cell to produce energy is compromised, and at the same time, the axon or the nerve fiber is demanding more energy. Okay.
So you mentioned both of you were researching the autopsy tissue. You were observing that. So is that a typical way when other scientists or other researchers are, observing the driving force, behind progressive MS, or like what actually chose you, or what led you to look at the autopsy tissue? Yeah.
So I guess it was partly my background, because when I did my MD PhD, part of my PhD was really looking at autopsy tissue. So that's sort of a kind of a natural progression. But there are other approaches, of course. You can look at non-invasive techniques. Kind of look at imaging. You can look at samples, so spinal fluid samples or blood samples, rarely sometimes you have patients where you're not quite sure whether they have MS or could that be a brain tumor or something unusual. So these patients end up having a biopsy, a brain biopsy. So that's another approach that one could take to look at brain biopsies. But also you got to bear in mind that these cases are slightly unusual. That's why they have the biopsy in the first place.
Right.
But so there are many different approaches. I guess the downside to looking at autopsy tissue is you're really looking at something right at the end stage, right? So you can't see the stuff that's disappeared. So you've got to be like a kind of a good detective to try and figure out what may have gone on at the early stages. So really the next step from the autopsy tissue is to develop models, so experimental models.
Right. It's funny that you mentioned the detective aspects, because it's like you have the end goal almost.
Right.
So you have to backtrack to see, what were the missing links.
Absolutely.
Right.
Yeah.
So, kind of basing off of what you and Dr. Nancy and the panelists are speaking about, how can, in your research in particular, how can the crucial role of damaged mitochondria help identify the new approaches to protecting nerve cells?
Yeah, absolutely. So it's kind of I alluded to early on. The therapeutic strategy in MS has been very much targeted at the inflammatory response and the myelin repair and so on. This neuronal compartment hasn't really therapeutically been directly targeted in a serious way. So this actually makes it very exciting to. So from a mitochondrial perspective, there's lots of neurodegenerative disorders where mitochondria are affected, and it's still an extremely challenge to identify treatments for, say, Parkinson's disease, ALS, and so on. The difference between progressive MS and the classical neurodegenerative disorders is that we get plenty of warning, certainly in the case of secondary progressive MS. So when someone presents with optic neuritis, there may be a decade or a couple of decades before they develop it, so we could really catch it early on. So I think, trying different therapeutic options, targeting the neuronal pathways and mitochondria within neurons, it's a very exciting way of trying to solve the problem.
Yeah, it's exciting in its own right.
Mm-hmm.
Yeah. And lastly, basically, I wanted to kind of touch on what Dr. Thompson had mentioned a little bit. He was saying how, the Alliance grant kind of serves as a catalyst to unite the top institutions.
Absolutely.
So using that train of thought, how do you think these global efforts are going to be, helping shape your own research now that, the grant, all this is coming along?
Right.
How can that global unification help your mitochondrial research and your vein?
Yeah, of course. So we've been collaborating with different international centres, over the last few years. So we've already formed some links which we could really utilise with a focus on progressive MS. And in addition, we're looking at kind of linking up and forming new collaborations to try and kind of facilitate this kind of the mitochondrial strand of work. So there are many aspects to this, is to try and look at more postmortem autopsy tissue to see is there anything that we've missed, or there's further work that needs to be done, and then really developing experimental models where you need a lot of kind of a basic science, strong background, and really disease relevant models. Okay, the collaborations could help a great deal in that respect. I mean, we've been working on these aspects for a while. Imaging is another aspect. We can bring in MR spectroscopy, for an example, to look at the brain metabolism. And finally, really, we're thinking in sort of a creative ways of how we could translate these findings in experimental models into patients with progressive MS. And we've got a couple of ideas of how we could put forward clinical platforms to link up with the pharmaceutical industries to really put the icing on the cake, if you like.
Absolutely.
Okay.
Well, thank you so much. And I wanted to just, on a last note, after this, after this meeting, after this conference, what are your immediate next steps in regards to the this research?
Yeah. So I think the next thing is really to get the team together. And really put an outline to see, what is it that we really, if we were to be offered the large sum of money that was just announced for the second round, what is it that we want to achieve? At the end of 4 years, how are we going to impact patient care? I mean, that is the most crucial question to me, and it's getting the right people. So that's going to be the right priority, really.
Absolutely.
Okay.